Hi, my name is Udi So, and I am an international business leader in J2K Bio. J2K Bio is a cosmetic ingredient manufacturer located in Korea, and we are specialized in microbiome fermentation and botanical extract with our unique technology. Today, I'm going to introduce our new cosmetic ingredient, Metabiome HK. Metabiome HK is produced from microbiome fermentation, and it is effective on skin irritation reduction by rebalancing skin microbiome. Before I introduce our product, I'd like to introduce a little bit about our research technology. Then, let's go! J2K metabiome is a compound world of microbial metabolites and microbiome. Microbial metabolites are intermediates or products of microbial metabolism and are usually small molecular compounds produced after fermentation. Microbiome is an ecological community of commensal, symbiotic, and pathogenic microorganisms in plants or animals. And human microbiome is the collective genomes of microorganisms living in human body. In J2K Metabiome Bioresearch Center, we isolate and identify useful and beneficial microorganisms from nature, and we develop an appropriate fermentation technology for each microorganism. Using our fermentation technology, we study about bioconversion of natural compounds after fermentation, and we evaluate their biological effectiveness on skin so that they can be used in skincare products as active ingredients. These are the list of microorganisms isolated and identified by our metabiome technology. Since J2K Bio was established in 2017, we have isolated and identified more than 200 species of microorganisms from nature and food, and we confirmed that about 26 microspecies are applicable to cosmetics among them. We are developing new cosmetic ingredients using these microorganisms, and we can also develop customized cosmetic ingredients for individual customers using these microorganisms. J2K Metabiome is using mainly two types of fermentation technologies. One is J2K Symbiotics technology. J2K Symbiotics technology is a microorganism culturing technology with prebiotics presence. Using our natural extraction technology, we extract beneficial components from nature and we ferment our microorganisms with those natural extracts as prebiotics. By this technology, we can obtain bioconverted active ingredients after fermentation, and the product will show higher effectiveness than general natural extracts. The other one is J2K DMS. DMS stands for Dual Microbial Symbiotic Technology. In DMS technology, we use two different microorganism species and we culture them at the same time or sequentially. By using DMS technology, we can obtain more effective active compounds than ones from single microorganism fermentation. Our Metabiome HK is produced by DMS technology. Now I'm going to talk about our product, Metabiome HK. Our Metabiome HK was born in Jeju Island of Korea. At the beginning of our Metabiome project, we focused on Jeju Island because Jeju Island is abundant in biological diversity due to its unique environment. And Jeju Island is one of the biggest green tea supplier in Korea. So we collected some green tea flowers and we isolated some microorganisms from it. Among those isolated species, we identified one Saccharomyces species and we named it as Saccharomyces sp J2K23. And the food culture of Jeju Island is based on soybean paste and traditional alcohol making yeast. So we also looked in Jeju Island traditional alcohol brewing yeast, and we isolated microorganisms and identified one Bacillus species and named it as Bacillus sp J2K51. Saccharomyces sp is an yeast species generally recognized as safe for human consumption. This species is generally used for winemaking and baking bread. This species is also widely used in food industry as the resource of vitamins and minerals. Bacillus sp is gram-positive bacterium also used in pharmaceutical and food industry. Also, according to some research papers, Bacillus sp can produce hyaluronic acid, which is used for moisturizing agent in skincare products. As mentioned earlier, our Metabiome HK is produced by dual microbial symbiotic technology, culturing two microorganisms sequentially. We first culture Saccharomyces sp J2K23 to obtain beneficial substances to prepare for second fermentation. And then we secondly culture Bacillus sp J2K51 in the culture medium of prior fermentation. By the reaction with the product of the primary fermentation, we could obtain highly effective biopolymer at the second fermentation. Our Metabiome HK is targeted to provide healthy skin by rebalancing skin microbiome. So I'm going to talk about skin microbiome and its importance in our skin health. And I'm going to talk about how our Metabiome HK works with those skin microbiome. In general, we understand that our body is composed with the skin, which is the largest organ in human body, bones, muscles, blood vessels, nervous systems, and internal organs. But we have one missing component, and that is microbial ecosystem, and we call it microbiome. I'd like to give you some facts about human microbiome. In a human body, there are more than 100 trillion microorganisms. Among them, microbes living in our gut weigh up to 2 kilograms. The microorganisms existing on our skin can be divided into two groups. They are commensals and transient. Commensals are no problem and even beneficial for host most of the time. However, transients are pathogenic and colonized after injury or trauma and may cause infections and inflammations. On skin, there are about 1 million microbes per square centimeter with about 40 different species. Human skin is generally colonized by beneficial microorganisms and serves as a physical barrier to prevent the invasion of pathogens. But when microbial ecosystem between commensals and transients is imbalanced, skin disease can result. So human immune system and commensal microbes communicate each other to preserve barrier function of skin from outside invaders. Although our skin is colonized by so many different types and numbers of microorganisms, each body site has different diversity of microorganism species. For instance, the majority bacteria in our forehead, where the skin is generally oily, is Propionibacterium acnes, followed by Staphylococcus epidermidis. In our foot site, however, the major constituent is Corynebacterium species, while Propionibacterium species colonized only a small quantity. Based on those research results, we could know that commensal microorganisms and their distribution are different according to skin condition. Under the healthy skin ecosystem between host and microbiome, host immune system interact with the commensal microorganism, and they help each other to prevent skin inflammation by regulating cytokine production, inhibiting pathogen growth, enhancing immune system, maintaining epidermal integrity, and etc. This slide shows general allergic dermatitis pathway. When the allergen attacks to normal skin, our immune cells, such as Langerhans cells, helper cells, and killer cells, are activated, and immunological molecules, for instance cytokines and chemokines, are produced. Due to the immune reaction, it causes skin inflammation and itching, and it finally results scratching of skin. Because of the scratching, our skin barrier function is disrupted, and our skin becomes more inflamed, resulting chronic skin dermatitis. This is a general chronic skin dermatitis pathway, but we have one important factor missed in this pathway, and that is microorganism in the skin. This slide shows the skin dermatitis, including the function of microorganism in the skin. When our skin is influenced by outside stress, such as allergens, accidents, or environmental factors, our immune system is automatically triggered to defend our body. By the foreign invasion at the same time, our skin microbiome ecosystem is also imbalanced. Due to this imbalanced microbiome ecosystem, our skin is colonized by pathogens such as Staph aureus. When our skin is colonized by those pathogens, commensal microorganisms, for instance Staph epidermidis, interact with the skin immune cells, and it helps to activate our immune system. In our defense system, there is a natural peptide called AMPs, antimicrobial peptide, and these antimicrobial peptides are secreted by commensal microorganisms and our skin keratinocytes. Secreted AMPs kill directly the pathogens, and it also boosts the activation of immune cells. When our immune system protects foreign invasion well, and our skin microbiome ecosystem is rebalanced, our skin can return to normal and healthy skin. Here we can see another example of the importance of skin microbiome in the acne lesion. When our skin is invaded by pathogens such as Staph aureus and C. acne, Staph epidermidis inhibits the expanding of Staph aureus, and it also inhibits the proliferation of C. acne. At the same time, Staph epidermidis induces keratinocytes to produce AMPs in order to kill Staph aureus. Then, what is AMPs? As explained previously, AMPs stands for antimicrobial peptides. AMP was first discovered by Alexander Fleming, who discovered penicillin. All living organisms uses AMPs as their host defense system. It kills directly pathogens, and it also helps to activate host immune system. Among AMPs, cathepsins and beta-defensins are the most well-characterized AMPs in human skin. Among cathepsins, LL37 is the only member of cathepsin family, so we also call it as cathepsin LL37. LL37 has antimicrobial, antifungal, and antiviral activities, and it is a mediator of inflammation, and it also connected with the re-epithelialization and angiogenesis. This slide briefly shows what kind of functions LL37 plays in our skin. Based on this background information, we studied about our Metabiome HK on skin microbiome rebalancing activity, focusing on its AMP expression promotion. Generally, during the in vitro test, only skin cells are cultured and their RNA or protein production were measured. But because we want to see the interaction between cells and microorganisms, we used a different cell culture system, which is cell and microbial co-culture method. First, we cultured keratinocyte HK T cells, and then we cultured Staph epidermidis with existence of cultured keratinocyte cells. In the co-culture system, microorganisms and cells may communicate each other, and they may influence each other, and they may produce different types of products during the culture. According to our pre-test result, we could confirm that keratinocyte HK T cells interact with the Staph epidermidis and produced little amount of cathepsin LL37 in the serum-free culture medium. So we conducted the same test with the presence of our product, Metabiome HK, in the cell microbe culture system. As we confirmed in our previous test, Staph epidermidis interacted with keratinocytes and produced small amount of antimicrobial peptide. But when we treated 1% of our Metabiome HK, we could see that remarkable amount of antimicrobial peptide was produced compared to our previous test without our product. So, when Metabiome HK was treated on keratinocyte cells in the presence of staph epidermidis, the expression of antimicrobial peptide were much increased, and this result suggests that Metabiome HK promoted the production of antimicrobial peptide, and our skin can have more power to insist from pathogen invasion, and it helps our skin to rebalance our skin microbiome ecosystem. In addition to skin microbiome rebalancing effect of our Metabiome HK, we also conducted the moisturizing effect of our Metabiome HK. For moisturizing effect, we checked the expression of aquaporin 3. Aquaporin is simply the channel of water for cells because it selectively transfers water molecule into cells. This is a test result of our Metabiome HK from aquaporin 3 mRNA expression. As the concentration of Metabiome HK increases, we could see that the expression of aquaporin 3 mRNA expression was increased in a dose-dependent manner. In addition, we also checked the water retention ability of our Metabiome HK by cut apple test. As you may know very well, when apples were cut and stewed, the water in the flesh is evaporated and it quickly dries out. We designed this test based on this fact. So we prepared 2 pieces of cut apples, and then we applied water to the left one and the same amount of the Metabiome HK to the right hand one. And then we let them dry out on the table. After 10 minutes, we put thin papers on the surface of the cut apples, so that the paper absorbed the water on the surface of the apples. And then we visually compare the amount of the water on the paper. As you can see in this photo, the paper attached on Metabiome HK retained more water, and this means Metabiome HK protected cut apple from dry out and helped to retain the water on the surface. In addition to moisturizing test, we also carried out anti-inflammatory efficacy test. The left one is nitric oxide production inhibition efficacy of Metabiome HK on mouse macrophage cell, and nitric oxide was induced by sodium lauryl sulfate. As you can see in this chart, nitric oxide production was increased after treatment of sodium lauryl sulfate, but after treatment of Metabiome HK, the production of nitric oxide was dose-dependently decreased. The right one is cyclooxygenase 2 production inhibition effect of our Metabiome HK. As you can see, cyclooxygenase 2 production was dose-dependently decreased after treatment of Metabiome HK. Based on those in vitro study results, we carried out a clinical test to see anti-irritation and skin soothing effect of our Metabiome HK. In this test, we prepared cosmetics with and without 3% of Metabiome HK, and we tested on 20 women aged between 20 to 55. Skin microbiome unbalancing and irritation were induced by 1% of sodium lauryl sulfate, and we asked the subjects to apply samples for 3 days. During the test, skin redness was measured by spectrophotometer. This is the test result. At day 0, due to sodium lauryl sulfate treatment, skin was irritated and microbial ecology was unbalanced. So, the skin redness was increased from 7 to about 12. As time goes by, the skin redness of both test and placebo sites were decreased, but we could see that the test site with Metabiome HK was 11.3% decreased after 3 days application of cosmetic containing our Metabiome HK. Based on this test result, we assume that application of Metabiome HK enhanced the communication between commensal microbes and skin cells, and due to increased communication between them, AMP production was promoted. Produced AMP quickly rebalanced the skin microbial ecosystem, so the skin redness at the test site was much improved than the placebo site. These are some cases after treatment. As we can see here, the skin redness at the test sites are remarkably improved compared to placebo sites. So this is the conclusion. Our Metabiome HK was born by our unique symbiotic fermentation technology. And we confirmed that our Metabiome HK promoted the production of AMP in the cell microbe co-culture system. So we assume that our Metabiome HK may help to rebalance the skin microbial ecosystem by promotion of AMP production. And we also confirmed that our Metabiome HK is effective on skin irritation reduction, and we assume that this anti-irritation efficacy of our Metabiome HK is by AMP production promotion and anti-inflammatory effect. And because Metabiome HK is a biopolymer, it has a skin moisturizing effect. Lastly, it has a strong anti-inflammatory effect as well. This is a typical property of our Metabiome HK. The inky name is Bacillus ferment, and 1,2-hexandiol is used to preserve the product. Metabiome HK is a colorless to very light yellowish and viscose liquid. Okay, thank you for listening to my presentation today. Please visit our website just for fun, and please also send me emails if you have any questions or requests on our Metabiome HK. You can also send me emails for personal questions as well. Have a nice day and see you later!